Hi everyone, and just like that, it's the final night of See and Learn 2022. We've got a great evening in store for you with our special speaker, Julia Notar, with a different take on urchins. And for those of you lucky enough to be on Saba, we've got a special menu with tasty soup to bring in the winter months, and pizza slices, and great drinks. And we are here at the Busy Bee, which is part of Breadline Plaza. And it's also the last night to buy raffle tickets. This is just a sample of some of the physical things. This doesn't show the incredible 3-night stays at different villas and private homes. And of course our grand prize, which is a week on the Caribbean Explorer Live Aboard, which is part of Explorer Ventures fleet. So join us tonight. We'll be back before long. Thank you.
All right, question number 4 is Gina Clementi, who specialized in moray eels. What special biological feature makes morays such successful hunters? Yes. Yes, in the back? No, not electric shock. 2 jaws. Yes, correct. And bonus question, what is the second set of jaws called? Jaws 2. I like that. I like it. Anyone? Nope. All right. Oh, where? Okay, there it is. Go back. The second smaller set of jaws is located behind the pharynx in their throats and is called pharyngeal jaws. Next question from Sheila Scalero, which specialized on seagrass. What feature distinguishes the invasive species Halophila stipulacea? Broadleaves. No. What's it? Veins. Yes, it's very veiny. Oh, he said it back? First? Said it first in the back. Yarno. Oh, you have a hat. You have a hat. Next question is about Vince Capone and his undersea mapping and sonar. Worldwide, how much of the ocean floor bathymetry has been mapped using sonar? This is a percentage. 10%, 5%, 30, 20. Who said 20? Do they already have a hat? He got 20%. Got a hat? Okay. Bonus question. How deep was the mapping crew able to collect data from in Saba in April? 1,400 feet. Anyone? Anyone else? It's raining. It's no excuse. It's online. 35 to down to 590 feet, which is 10 meters to 180 meters. And they collected over 120 million data points. Next question. The snails from Tello-Neckheim, all about land snails. This is going to be a great question. When a male and female snail meet, how can you tell the gender? Exactly, yes. You can't. They're hermaphrodites. They somehow decide. Oh my God. Kind of a trick question. Next question is from Lowell Iparak, and he was presenting on sargassum seaweed. Yes, don't open the Ziploc. That's not the question. The question is, out of the 300 and more species of sargassum, how many create the blooms or influxes?
3.
No. 2. 2 is correct. Sargassum fluitans and Sargassum natans. Oh no, the beer is spilled. Oh, that was mine. Rude. Okay, bonus question. Sargassum originates from the Sargasso Sea, but where is the second Sargasso Sea that he was talking about located?
North America.
Brazil and Africa. Sally's got it. Next question from Susan Perkins, our anole expert. Although both humans and anoles can contract malaria, are humans able to get malaria from lizards? No, you all get hats. Just kidding. No, most malaria parasites are quite specific to their hosts. And from Dr. Aaron Muller, who presented on stony coral tissue loss disease. Coral scientists and pharmacists are teaming up to fight against Skittle D. What is the efficacy rate of the antibiotic treatment? 80%, yeah, 84%. And from Florentine, who is here in the audience, she talked about magmatic evolution. Her question, she studies plagioclases to study the evolution of the magmatic system over time. What is a simple definition of a plagioclase? It's in rocks. Crystal, yes, Melinda. And a bonus question. She said that both our underwater and topside hot springs are an example of what? Did someone guess it? Passive degassing? Who said passive degassing? Nobody? No hats. I get a hat. All right, Elwin did a bonus presentation on sea urchins. Is Elwin here tonight? No? Yes? Most important question from his presentation. What caused the 2022 diadema die-off? Bacteria, yes. Does anyone remember? Ciliates. Yes, correct, ciliates. And from our most recent presentation by Lydia Mathker, who studies animal pupil shapes and coloration. Why are animal pupils shaped the way they are? Is it A, body shape, B, lifestyle, C, habitat light field, or D, eye optics and processing? Okay, I'm hearing a lot of answers. The correct answer is, oh, we didn't do a slide. The answer is all of the above. Good job, Michael. And now our speaker for tonight is Julia Notar, who will be presenting on sea urchins as well. And I will hand the mic back over to Lynn.
Well, that was fun. Which one is it? The big one? Okay, one more time. We got to go through our slides. Let's hear it for Carib Trans. Thanking for our live streaming. And when we say that, probably equally as important has been the Makana Ferry and Saba Sea Transport, which brought a number of our experts here. Brought a number of our experts here complimentary. So let's also hear it for the Makana and Saba Sea Transport. Okay, tonight's speaker is sponsored by Hibiscus Cottage. Hibiscus Cottage is on the path that we call, geez, I know this. Kai, what's your street? Sorry? No, it's not the Museum Street. Anyway, there's a nice name for it. Anyway, no, it's a beautiful 2-bedroom, 2-bathroom cottage, and people like it because it's an easy walk. But they have been very gracious sponsors, and thanks to them for sponsoring Julia, who can tell you also it's a nice place. Our work would not be possible at all in this program without the support of public entity SABA. We have some representatives there from tonight. Vito was just here, so thank you both for the support. And Prince Bernhard Culture Funds is who pays a small stipend to our professional experts who come in, which helps defray their travel costs. So thank you to Prince Bernhard Culture Funds. And then we have our many, many sponsors. Many are here in the audience tonight. We've got Larry, Sherry, we got Michael from Ira's house, we've got Debbie and Terry, the Saba Conservation Foundation, help me out, Marie with her nice bracelets and raffle prizes. Raise your hands. Oh, John, who made a knife. Andres for El Mamo Cottages. The Busy Bee Bakery. And, am I doing that? Okay. Anyway, but there's many sponsors that make this possible. Thank you very much. One more time, last chance to get these great raffle prizes. We'll keep selling tickets until you make us stop. So tonight, the big reason we're here is to introduce our speaker. So what do sea urchins and brittle stars have in common with Scarecrow from Wizard of Oz? If I only had a brain. They don't have a brain, but that is no problem. And Julia Notar is here to tell us how these animals can still see and learn. Julia earned her PhD in Sanka Janssen's lab, and we had him here as a great presenter previously, where she studied the visual ecology of marine invertebrates. She's interested in animals with distributed sensory and visual systems. What can these animals see, and what decisions do they make with the information they can gather from the environment? Her research mostly focuses on sea urchins and their visually guided behavior in a comparative context. While she primarily works with urchins, she is also interested in other echinoderms, marine invertebrates, and distributed and low-resolution visual systems more broadly. How's that for a mouthful? Let's finish out See and Learn 2022 by looking sharp with Dr. Julia.
Oh, sorry. Oh, can I have the clicker? Hi, everyone. I need the clicker. Thank you. Hi, everyone. Thank you so much for joining us, and thank you to the folks from See and Learn who invited me here. Thank you to everyone from SABA who I've met. You all are an absolute pleasure, so I'm really thrilled and honored to be the final speaker for this series. You know, no pressure. And as stated earlier, I'm going to tell you about some of my dissertation research on sea urchins and brittle stars, and I'm going to talk about their vision and the fact that they are able to learn. Now, I like to start out by just appreciating the wonder of the natural world, and this is not, I think, a point that I have to sell too many of you here in the audience on, but the natural world is totally diverse. It's very visible if we want to look at the beautiful colors and patterns around us. It's also full of things that just absolutely defy convention, urchins being one of them. And it's just absolutely beautiful. So I hope you'll enjoy some of these themes as we go through this presentation. But speaking of things that defy convention, I think this is what my parents thought of me when at age 6, they asked what I wanted to be for Halloween, and I said banana slug, which is this nice yellow slug found in some of the forests in California. And 30 years later, I'm still dressing up as yellow slugs for some reason. So my love of unconventional and slightly strange animals really runs back a long time before I even knew that it was going to point me towards a career in science. So in looking at vision and the vision that humans and animals have, if we think of a spectrum of vision where we've got sort of more normal vision here on this side, we've got animals with 2 eyes like we have 2 eyes. There's this sea otter up here, this little bobtail squid, and the mantis shrimp all have 2 eyes. That seems pretty conventional. And there's also lots of animals that have lots of eyes, so many that, you know, we'd have to get out most of our fingers and toes if we were going to count them all. So we've got this scallop down here. We've got a lovely tube worm. If you see these spots on the tube worm's fans, those are the very small eyes. And then on the top, we've got chitons, which are kind of like a flat snail, and they've got eyes distributed all through their back that actually go through their shell. And then as we continue on our journey of vision, we get over to the extremely unconventional side of this spectrum where we have sea urchins and brittle stars that I study, and they have no eyes. So without eyes, you'd think that they can't possibly see, but the evidence that we have shows that they can. So here's a video taken from snorkeling underwater, and you can see that as the snorkeler approaches these sea urchins, they're waving their spines in a visual response to the presence of the snorkeler. Anybody diving, snorkeling here, or even in the tide pools, seeing some of the diadema do this locally? Yeah, it's a very, very recognizable response. And so in the lab, I can do experiments like this, where we set up our urchin in a little urchin movie theater, and if you look at the top of the screen, you'll see a black dot appear on the little TV screen I've put there before the urchin. And this is going to repeat, and you'll see the urchin wave its spines in response. So this is one of the ways that we can do experiments in the lab where we're definitely controlling everything that we can. We can see that it's just a visual response, not one that's related to water movement. So a question that I get a lot is, why would a sea urchin need to be able to see? It's covered in spines. My God, those spines are so sharp. Why would it need to be able to see? It's well protected. Well, I think, again, if you've been out diving or snorkeling, you might have noticed that fish absolutely love to eat sea urchins. They're basically a nice little ball of energy-rich gonads. So if you've ever eaten sea urchin, you're eating the roe or eggs of a sea urchin. And especially reef fish, like the sheep's head here, that have really strong jaws, they can bite through the skeleton of a sea urchin. So we've got a clever little urchin hiding in a rocky crevice there, and it's a lot more protected because rocks are stronger than spines. Now, it's hard to overstate just how much fish love sea urchins, so I was trying to come up with a cross-cultural comparison, and I was struggling, so I just went with something that I love eating where I live in North Carolina, which are deep-fried Oreos. They're nice and crunchy on the outside. What's inside, delightful. And I think even if they were covered in spines, I would definitely pick through the spines just to get to that deep-fried Oreo inside. They have them at the state fair. They have them at gas stations. Yeah, North Carolina is a great place. North Carolina in the United States. I'm sure they have them other places. But yeah, if you go to the Sheetz gas station, they have deep-fried Oreos on the menu. All right, so when we want to think about how good is urchin vision, we can actually simulate this in the lab. So on the left here, we have a picture just taken with a camera of a reef, and this is what our vision is like. And one way we can measure the sharpness of our vision, or the visual resolution, is looking at it in degrees. Basically, how small or big of a detail can we detect? So for reference, 1 degree is, if you hold your thumb out like you're giving yourself a thumbs up, the width of your thumb is about 1 degree. So we have some of the best vision in the animal kingdom, and we can see it's 0.007 degrees. When we simulate what we think a sea urchin's vision might look like, we can see that it's a lot worse. It's 9.5 degrees. That's a huge difference, but you can still see some key features of the reef here. So even though it's pretty poor by our standards, it's still potentially very useful. So if you look at this picture, you can see that you can still see the outline of the reef. In the top right corner, you can still see that bright streak that shows the edge of the reef towards the surface. And you also have some places where you can see dark crevices in the rocks, which are, again, great places for urchins to hide. So just a little bit of a primer on vision so we can better understand how urchin vision works. I'm going to talk about different sorts of eye designs in the animal world. So the essential problem of vision is determining where light comes from. So if light is waves or photons bouncing around, we've got to determine where that's coming from in order to be able to detect an edge. So this is my PhD advisor with grad student of the year, running 10 years now. 1 year I'll win it. But the way he describes it is, imagine you're standing near a campfire with your eyes closed. And so you could tell what side of your body the campfire is on, but in order to pinpoint exactly where that fire is, you need a little bit more information about exactly where those heat waves are coming from. So if we think of light waves instead of heat waves, there's a few different ways that eyes can discriminate where waves are coming from. One is to use a lens. This is what our eyes do. This is what a lot of animals with 2 eyes do, but also the scallops. This focuses the light to a point. Another eye design is the compound eye that we see in a lot of insects, a lot of arthropods, shrimp, crabs, lobsters. And this sort of uses, like if you imagine an ice cream cone that you could catch raindrops in, they've got a bunch of those pointed in different directions around the environment. Sea stars and the fanworms also have compound eyes. There's also a pinhole eye, which works much like a pinhole camera, and this is what a nautilus uses. And then when we get to sea urchins, we actually think that their vision is based on the fact that they have spines. So if you look at these blue triangles here, these are simulating spines sticking out from the body that would block light from other directions. So given that sea urchin skin is very sensitive to light everywhere, it's the spines sticking out that are able to block light from off-axis areas. This diagram on the left, I think, helps show it a little bit more. This is a skeleton of a sea urchin seen from the top, and you can see that that patch of skin in the middle of those spines can only see one part of the environment. A good metaphor for thinking about this is if you think about standing on the floor of the forest and you're looking up through the trees at the sky. If the trees are very far apart, you could see a large patch of sky, and if the trees are close together, you could only see a small patch of sky. So if we think of the spines like the trees in this metaphor, we could say that spines close together might give us smaller patches, which would be like smaller pixels in a camera, and so you could see more. You could see more detail, basically. Now, I get a lot of challenges on this idea about the spine, so one of the projects I decided to do was build a sea urchin-inspired camera. So most of the cameras that we have, use lenses and operate like our eyes. So I decided, why not build a camera that's like a sea urchin eye? So I built a 4x4 array of little light-sensing photocells that just sense brightness. We can record what they see, and if we add spines to the system, we can see how spines affect the system. So we built this, and then we would show them different arrays from a standardized distance to see if they could detect changes in light and different images. Now in reality, it didn't look as nice as this diagram, but this was a great pandemic year project when you're stuck inside. You can see that a lot of scientists turn to materials on hand when things get desperate. You can see some takeout chopsticks and some drinking straws there on the right acting as our spines. But when we look at the results from this, what we see is the spines really do improve resolution of the image. So on the left here is our system without any spines, and as we add 9 spines and then move up to a total of 15, that light square in the center is more discriminable. In addition, if we show it something else, just a dark square in one corner, we can see as we move from no spines up to a total of 15 spines, looking up there, that dark square, again, is much more discriminable. So this provides a nice physical proof of what could be happening with the sea urchins and their vision. So I'm going to tell some stories tonight about 2 aspects of vision that I've looked at in sea urchins, namely resolution and sensitivity. So resolution is some of what I've already talked about, sort of the sharpness of an image or the ability to determine spatial detail. And sensitivity has to do more with like the light required for an eye to function. And there's a general physical trade-off between these two. So imagine that you had a soccer field and you were watering it and you wanted to make sure the whole soccer field got equal amounts of water. So you wanted to set out containers to catch these drops of water from the sprinklers. If you used party cups, like here on the left, you could get a really, really fine scale idea of exactly where every drop of water was falling. But if you needed to have, let's say, 10 drops of water per container to know, if any water is getting in there, you might want to use these kiddie pools here on the right, even though you'd be losing some spatial detail as to where that water is falling. So if we think instead of water droplets, we think photons like light, eyes kind of have to trade off between catching enough light to create a signal and having enough detail in here to determine exactly where details are coming from. So today I'm going to tell you about 3 different projects, 2 about urchin vision, looking at resolution and how it differs between different species, 1 about sensitivity and looking at whether the urchin is sensitive to light equally on all parts of its body, and then finally I'm going to tell you about how brittle stars can use light information to learn. So sea urchins live in a whole variety of habitats worldwide. There's about 1,000 species in the world, and they're found all the way from the tropics to the poles to the deep sea. And this means that they live in all sorts of different visual environments. So if any of you saw Lydia's talk a couple days ago, she talked about how animals with different pupils can be adapted to different lifestyles or environments. And we can look at this in sea urchins as well. So when we think about how these different aspects of where different species live, they can break them down into categorical environmental variables. So we can ask, do they live in bright or dim environments? We can ask if they live in visually simple or visually complex environments. And we can look at the temperature of the water, because vision usually requires a very high metabolism. We think that maybe species that live in very cold waters might not be able to see because their metabolism just isn't high enough. So we want to understand if their spine density, which again we think determines resolution, varies by visual habitat. And we have to also take into account whether or not these animals are closely related. So I looked at 33 different species, and we took photos of these animals, and we quantified the dot patterns that their spines create. So on the right, you can see the dot patterns corresponding to each species, and you can see that some have very, very small, packed spines, and some have spines that are very far apart. So we're able to quantify the distances between these spines and estimate resolutions. So when you look at a whole range of estimated resolutions for urchins, we find that our best urchin actually does pretty good. I would be happy with that if I had to swim like that underwater. Our worst urchin doesn't do great, but it's trying. It can at least still see that corner of the reef. Now this is a big science diagram. I'm going to walk us through this real slowly. But we can put all these 33 species together on a tree that shows the relationships between the two species. So what's important to know is that when two species have two branches that are close together, they're closely related. And then the next most closely related ones would be like cousins and so on. So don't worry too much about that. But what we can understand is that these blue circles show basically how big or small an object would need to be for the animal to see them. And then we can add on top of that, that layer of those habitat variables and those categories. So just to take you through a few groups that pop out right away, we have a group called the slate pencil urchins that have really wide, very widely spaced spines, and they live in tropical reefs or the deep sea. We have a group of deep sea urchins that have really, really small spines that are usually very closely packed. We think they might use them for locomotion mostly. And then we have a large group here of what I like to call the urchiny looking urchins because they're the most regularly shaped, they've got the most regularly sized spines, and they all have resolutions that are about on par with that average urchin resolution that I showed you. So the big takeaways from this are basically that the relationships between these species end up mattering a lot for the density of spines on the body. And we found from our analysis that it turns out visual habitat doesn't matter as much. So as opposed to other animal groups where we usually see that the eyes are well adapted to the environment, when it comes to visual resolution, we don't see that in the urchins. So that's a little perplexing. But when you think about it, spines are used for a lot more than just this visual process. You know, I talked earlier about how much fish want to eat urchins, so spines are primarily used for defense. They're also used for the animals to move and walk around a little bit. So we think that maybe the spines and the vision here is actually sort of a downstream process from what the primary responsibility of the spines are. So urchins, full of surprises. This was the first one on a really cool journey. And now we're going to move on to talk about the next part of that journey, which is looking at sensitivity across the body. So the sensitivity of vision can determine a lot of different things about an animal's lifestyle. So for example, it can vary by habitat, it can vary by time of day and diurnal cycle. These bright blue spots here are bioluminescent flashes from ostracods or little tiny crustaceans. They use these flashes sort of at crepuscular nighttime periods to signal to each other in little mating dances. And also, behavior and posture can influence sensitivity. This looks like a pile of leaves and rocks and shells, but the urchin attuned among you will see the spine sticking out from the bottom. So this is actually a white urchin that are found sometimes around here, and it's covered itself with leaves and shells, maybe for protection, but also to shade itself from light potentially. So I wanted to ask, is a sea urchin equally sensitive to light all over its body? If you're a 360-degree eyeball looking in all directions at once, is your vision as good in every direction at the same time? Or are there specializations? So I looked at a few different regions of the body. First of all, I wanted to compare two regions, the ambulacrum and the interambulacrum. You don't need to remember those names, just remember that one is very full of sensory structures, and the interambulacrum is the in-between those areas region. So this ambulacrum, which you can see here on the skeleton, has a lot of holes in the skeleton, and this is where those tube feet the structures that animals use to move around, go through the skeleton. It's also where all of the major nerves are. So this is a prime candidate for a very sensitive area because it's got a lot going on there. The regions in between, they don't have any of those structures. They don't have the major nerves. So they're a lot less likely to be sensitive. I also wanted to ask, as you move up the sides of the urchin from horizontal to overhead, does sensitivity differ? These are parts of the body that view different parts of the environment. So I'm going to show you a couple of videos now about what these trials look like in the lab. We put urchins in sort of basically a little corridor and we shine a light on them. And here's an urchin moving away. This video is sped up quite a lot. You can see the urchin is like, nope, don't want to be near that light. I'm getting away, thank you very much. So this is what we call an away behavior. And we also see the urchins sometimes moving towards the light in this video. So in this one, the urchin, for some reason, decided the light seemed delicious, climbed on top. I like to call this total eclipse of the urchin because it sort of blocks out the sun. And if we quantify, how many animals are moving away and how many are moving towards, we can come up with a map of sensitivity, depending on what region of the body that we're looking at. And in summary, we find what we expected when we compare those two different regions of the body, that the one with all the sensory structures, all the nerves, is very sensitive. And the one without the nerves and without those sensory structures is less sensitive. So even though we expected that, that's great to be able to confirm that. When we look at this on a living animal instead of just the skeleton, we can see these white structures here. These are the tube feet, and you can just see how packed they are in those ambulacrum regions, and that they're not there in those in-between regions. When we look at that elevation on the body, moving from horizontal to overhead, we find that on the horizontal plane, the animals are less sensitive, but they are most sensitive to light above them, which is interesting. And I think of this in an ecological context. Again, we're going back to habitat and where the species lives. And this species predominantly lives in seagrass beds. So this is what we would call a horizon-dominated environment, in that you're basically, determining there's an overhead and then there's your horizon. So our thought was that being less sensitive on the side of your body can kind of help you monitor, if you're getting close to the edge of the seagrass bed, you don't want to move out of it because then you're going to be away from all your food. But being more sensitive overhead would be beneficial for detecting things like fish, which would mostly be coming from above. So we find this interesting difference in sensitivity on different parts of the urchin body, which indicates kind of that you've got different parts of the eye doing different things at the same time, which is pretty cool. Unconventional, but cool. All right, let's move on from talking about sea urchins, and I'm going to talk about learning now. And this is where the brittle stars sneak into the picture. They're a lot faster moving than sea urchins, so this is why I decided to use them instead of urchins for this learning project. But I think these results are sort of broadly applicable to urchins as well. So a little primer first on echinoderm nervous systems. As mentioned earlier, they don't have a brain. So they're a bit different from our own nervous systems, which we have using a centralized nervous system. We think of our brain kind of like a computer on top of a stick. We use the stick parts of our body to get around, and then all the information that we gather goes to the brain to be processed and then sent back out. On the other hand, echinoderms, so sea stars, brittle stars, sea urchins, sea cucumbers, their nervous plan is different. It's basically all periphery. There's no centralized computer. So we can see in this diagram of a sea star that there are 5 nerve cords that run down each of the 5 major regions of the body, and they're joined in the center by a ring, just by a circle, not a brain. And this ring doesn't seem to have any processing capabilities itself, but it lets these 5 different nerves talk to each other. So we've sort of got a committee of 5 here discussing and making decisions. So can a nervous system like this even learn? It's so different. So there's really not much studied here. There's a couple of studies on sea stars themselves that show that they can learn, but I think, since 1901, I think there's about 6 studies on this, which is, for scientific reference, not many at all. So I wanted to look at brittle stars, which people haven't looked at yet. And I used this species here, Ophiocoma echinata. And I wanted to know, can they learn to associate a period of darkness with a food reward? And to set up our training protocol, I'm going to use a metaphor here. Imagine you're at home and you hear some music outside. You might not do anything. You might not respond at all. But the next time you hear music, if you see the ice cream truck outside, you might decide to respond. And you run outside and you get your reward. You get a treat. You're very happy. And so you might form that connection in your brain between ice cream and music. So in the future, when you hear that music, you may remember that connection, and even if the ice cream truck isn't there, you respond. So this is how I set up the way that we're going to test our brittle stars. So instead of using music and ice cream trucks, brittle stars don't really interact with those things very often. We're going to use something they love as much as I love ice cream, which is a piece of shrimp. They go nuts for it. And instead of music, like I said, we're going to turn the lights off. We're going to use a period of darkness. So we turn our lights off, and we present the shrimp, and the brittle stars run out and they eat the shrimp. Good stuff. That's how we train them. So to test them, just like in our ice cream metaphor, we turn our lights off. We hope something's going on inside there. Fingers crossed. And we want to see if the animals respond even when there's no food around. So this is our training and our testing setup. So I'm going to show you some videos that are sped way up just for reference. These are much faster than these animals actually go. But I'm going to show you some videos of them eating and showing how we train and test them. So this is before the animals have any training at all. This is just feeding them under the normal lights. You'll see me put the food in, and the animals crawl out from behind the filters where they hide. They move over to where the food is on these pipettes, and they pull the shrimp off. Then they run back behind the filters and they eat. Very sneaky. All right, now I'm going to show you how we train them. You'll see the lights in the room turn off, and we've lit the room with dim red light, sort of like people do with a dark room with cameras. These animals are not very sensitive to dim red light. So you'll see the lights go off, you'll see me put the food in, and again, the animals are going to come out and eat that food. So as soon as the food's in there, they're coming on out, they're searching for it, they manage to find it. And so we repeated this about 14 times to try and get the animals to form that association. Oh yeah, we'll see in the data in a little bit. There are definitely some fast learners and some slow learners, absolutely. All right, so this is where the magic happens. I'm going to show you a video of one of our testing regimes. So this is an above view instead of a side view. And again, you'll see the room lights go off. I haven't put any food in the tanks yet. And you'll see animals in these 3 central tanks across the middle here move out. So the lights go off, moving at 60 times our normal speed, but things are still a little bit slow in brittle star world. And you'll see this animal in the center move out. So again, there's no food in here at all. And then you'll see this animal in this tank kind of move into the middle, and this one moves into the corner next to the filter. This is me coming in and putting in the food, and then you see all the animals move out. But you can see how 3 of the animals responded even before there's any food in the tanks. All right. Scientists love movies, but we love data even more. So I'm going to show you some of the data from this project. So we started the beginning of our project, and the days of our project marched forward for the entire year that we ran this. And for every time that one of our animals that received training comes out, you're going to see a red square. And we have an untrained group for comparison. So every time one of those animals comes out, you'll see a black square. So when we run our control period, like that first video, the animals aren't receiving any training. And they don't come out if there's no food when it's dark. So that's a good baseline to have. When we train them, this is when our trained animals are receiving their training. We see that they come out almost all the time, which is great because that means that they're forming that connection. And then when we delay in putting the food in, these are the animals that come out before there is any food in the tanks. So you can see that the trained animals, because they received training, they actually come out much, much more than the occasional sort of random times that the untrained animals come out. And as I mentioned, we've got some fast learners up here. The second row from the top up there, I call that our superstar. Just, it did so good. It came out every day. I wish I could have fed it more. But so we can see from this that the animals are emerging after that training, which is super, super exciting. It shows that they can learn. So to wrap up these 3 stories, I'm just going to talk about what I talked about. So in vision, we found that resolution varies widely across sea urchin species and doesn't seem to correlate with habitat. Oops. We talked about sensitivity. We found that sensitivity differs on different parts of the sea urchin body. And I showed you that brittle stars can, in fact, learn, which is really exciting. And to sort of wrap this all up and bring it to a close, one of the things I love about studying sea urchins is that no matter where I go in the world, as long as we're on the coast, there's a local species of sea urchin, often multiple ones. And people love to talk to me about sea urchins, even if they're like, I hate them, they're so sharp, So it's really incredible to get to connect with other people about sea urchins. But I want to make the point that our connections to animals and other parts of the natural world are connections to other people throughout time and space. So here's an octopus face from ancient Minoa. This is thousands of years old. Octopus has a center stage, but there's a sea urchin. Whoops, right there. People have noticed these things and lived with them for so long. And then here's a couple of more contemporary examples. Here's a mola from Panama and a sea urchin bark cloth painting from Hawaii. So that's really my big take-home message, is that animals connect us to each other in all sorts of ways. So I want to thank you. I'm ready to take questions, and I hope you'll agree with Dr. Cobb here, just sort of talking about how strange sea urchins are. The golden rule in echinoderm biology is that the obvious explanation is always the wrong one. Thank you so much. Are there any questions? Okay, I see a hand over here. Yes, sir.
Well, because your testing phase was very long, I was wondering, shouldn't they also learn in the end not to come out again?
Yes, so is the mic good? Can you hear me on the mic? Okay. So the question was, because the testing phase was very long, shouldn't they learn not to come out in the end? So one of the details I didn't share about that study is actually that big middle portion of that testing phase. We were actually still feeding them halfway through the period of darkness. So our period of darkness was 30 minutes. And for most of the testing phase, we gave them 15 minutes at the start of that dark period to show if they emerge. And then at the 15-minute mark, we would feed them, so they had 15 minutes in the darkness to still eat. So that would maintain that connection there. So we ran that phase quite a long time to sort of keep reinforcing things. And then we gave them a 14-day period where we moved the feeding to the very end of the dark period. So we had 30 minutes of darkness, and then we turned the lights back on, and then we fed them. So that was, again, supposed to sort of give them time to respond, so they've learned, still maintain some of that sort of time coupling between the two stimuli. And then at the very end, we had about a 10-day extinction phase where we went back to feeding them, 6 hours later in the light. So based on the protocols of the experiment, I would have loved to run that extinction phase a little bit longer to see, basically if that behavior did disappear. Just unfortunately with access to the lab issues due to the pandemic and everything, we weren't able to run it any longer. But yes, you would expect them to stop responding after basically receiving enough negative reinforcement. Yeah. Other questions? Was there one in the middle? Okay. Yes, right here in the front. Oh my gosh. Favorite question. I always get this question. Do they see colors or not? We don't think that they do. I did a small experiment on this at the start of my PhD. I didn't get very good results. It doesn't seem like diadema are sensitive to green. So if anybody wants to test other colors with other species, please go for it. I would love to see the results. One point I want to make about color information, it can give us a lot of useful information in the world. I prepared slides for this question. I'm so excited. So this is that colorful reef I've shown you many times. And if we take away color information from one half, we can still see so much spatial detail. We can still see a lot of important information. But instead of taking away color detail, if we take away brightness detail and instead make all the colors the same brightness, we get this picture instead. So this is making all the colors the same brightness. And it's actually way harder to tell what's going on. So I think this is just my personal hypothesis. I think this is part of maybe what's going on with the fact that animals have so many different kinds of color pigments and some can see lots of colors and some can't see a lot of colors, is that brightness information gives you a huge chunk of information to begin with, and color adds on top of that. But like I said, brightness just gives you so much information there. Thanks for a great question. Yeah. Yes?
Are the brittle stars primarily diurnal, nocturnal, or something else?
Yeah, so the question is, are the brittle stars primarily diurnal, nocturnal, or something else? So the species that I was using are primarily nocturnal. They actually do compete with other species that live on the reef near them for small rocky crevices. So during the nighttime, they'll kind of come out in the natural environment, they'll come out and forage, and when dawn starts, all three species will kind of move back to the reef and compete for the different crevices in the rocks. And so it seems like some of the species outcompete other ones with getting the better places to hide and the worst places to hide. The species I was working with is sort of, they were lowest on the totem pole in that, so they end up with the worst places to hide. But they are primarily night active, which was one of the reasons it was important to run a control and just see, before they have any training, do they emerge in the darkness at all? Which it seems like they don't when you just give them half an hour. Yeah. Other questions? Yes, in the middle.
Why do you think the brittle stars don't have eyes, but sea stars do?
So why do we think that brittle stars don't have eyes, but sea stars do? So people have been able to identify very small compound eyes in sea stars. They're basically at the ends of each arm. They've got a modified tube foot, that has a really, really small compound eye on it. So people have actually found those in sea stars. People have not found those in brittle stars or sea urchins. So I didn't discuss the research that's been done on brittle star vision, but it looks like they're sort of creating pinhole cameras on their backs with the migration of the pigments in their skin. So when their pigments are sort of aggregated at night, they can't really see, but when they're spread out during the day, it can kind of act like a pinhole camera, and they're able to resolve images that way. Their vision is about as good or worse than the sea urchins, but yeah. So there's also a brittle star vision story there that is a whole other exciting world. Was there a question in the back there? Yes.
Are they poisonous or venomous?
Oh, are they poisonous or venomous, the sea urchins? Oh God, I gotta remember this one. This is, put me on the spot. So I think venomous, the definition of venomous means that the animal is actually injecting a toxin into you. Poisonous is a more broad term. It means that it's, a chemical that irritates you if you ingest it or get it in your skin. So the sea urchin spines that, get stuck in your skin, they're usually they feel kind of itchy and irritating. So I would say probably more mildly poisonous, but they're not actually injecting venom into you. Yes, one more question there.
So if they can detect photons, they must have then receptors for it, right?
Yeah, so the question is, if they can detect photons, they must have receptors for it. Yes. So even though they don't have organized eyes, the basis of an eye is a photoreceptor cell, which receives photons. So there's been a little bit of work on looking at structures in the skin and cells in the skin and expressions of proteins that are associated with photoreceptors. So some of that work has showed that there are photoreceptors in different parts of the skin and some parts of the tube feet. So if you want to get into the nitty-gritty of it, there's different proteins that are associated with light that they've localized different ones in different places, but the moral of the story is that if you look at all of those kinds of proteins, they're all found everywhere throughout the skin. But they seem to have cells that look like photoreceptor cells in some parts of the tube feet, but those haven't been shown to be expressly functional yet. So there's a lot more work that needs to be done there. Yep. Yeah, I think that's it. Thank you all.
Anybody last minute raffles? Okay. Yeah. Everyone, once again, give a round of applause to Julia with that great presentation for tonight. And finally, the moment everyone has been waiting for, the raffle pulling. How exciting. Okay. Oh, we got two more. So this is the last chance. If you want to buy any more, this is your moment.
Last call, last call. Yeah, of course.
We'll shake them.
Thank you.
Okay. So here we go. The first pulling is for the Sabin Spicy Orange Liquor from Sabin Rock Living, a rum-based taste exposition of traditional Sabin fruit, herbs, and spices. Let's get to the bottom. Let's mix it up in here. Okay. Tadzio?
Yeah.
Tadzio? Are you here? Oh, okay, well, you won the bottle of rum, Tadzio.
Write it down. You gotta write. There, here's the pen. I can write it. All right.
Right here. Okay, so number 2. Get inspired, everyone, because there's 3 free entries to Busy Bee 5K run.
So 3 different poems, right? 3 different poems for the 3 entries.
Run or walk.
Okay, so we're going to do 3 pullings for the 3 different entries. Number 1. Owen. Woo! You get Owen. Yeah. Oh no, Owen. Lena's son. Odin. Yeah, I wrote it down. Odin. Sorry, Odin. Okay. Number 2. Owen. So is it a voucher? Duco, get those running shoes ready. Okay, and this is the 3rd and final entry for free 5K Busy Bee race. Ellen Ryan, Life Center, Ellen Ryan. Okay, and our next prize is, let me see. The 3D model of Saba. This is created from the mapping project in spring 2022, thanks to Aeronax Expeditions. They're really, really cool. Okay, let's see. This is a lot of pressure. So there's 2 of them, so we'll do 2 different pullings. The first winner is Claudia Every, another Life Center. Woo, give it up to Claudia. And the second 3D model goes to Christina. Angelina's Bioaches. Christina. Yeah. Okay. The next prize is 3 romantic nights. You can add to your reservation if you'd like to make it longer. At the new luxurious penthouse suite at the Cottage Club Hotel on Saba. This is a good one. he wants us to scoot over to the left a little bit. Oh, well, no, both of us, yeah. All right. The winner, Duco again. Go Duco. Congratulations. Yeah, Duco got a couple of. And the more nights you add, the longer your stay. Okay. The next prize, 2-course dinner for 2 with a bottle of their house wine from Saba's Only Wine Bar at the fabulous Shea Booba Bistro located at Lambie's Place. Let's see, dinner for 2. Duco! No. Eva DeVries. Woo! You're a winner, you're a winner. Date night, exactly. Congratulations. The next prize is the beautiful Eco Dyeing Scars by Anne Klein. Clean. Tadzio again. Tadzio in St. Martin. You're winning prizes tonight. You're lucky tonight. Next prize is handmade bracelets by Marie de Saba. There's 4 different bracelets, so I'm be doing 4 pullings. Duco! Guys, I promise I'm shaking it up. Duco's just very lucky tonight. Duco, you can give that bracelet to someone. All right, the second bracelet, Odin! This time it's spelled correctly. All right, the third bracelet, Odin! Mix it up. You do the next one, because I can't. Okay. The fourth and last bracelet goes to
Melanez.
Melnez. He's a student at the high school. Go Melnez. Okay, these are the 4 bracelets. 8. The next prize, Saba's new coffee table book, Sea to Scenery by Peter Johnson and Angus Martin. You do it.
What is it? Oh, Trisha, right?
Yeah.
Trisha.
Congrats. Okay, next prize. Ooh, this is a good one. Sunset cruise for 2 on Lambada. Oh, wait. No. Sorry. Learn something new or improve your game with tennis lessons from Lynn. A good one. A really, really good one. Larry. Nice. Okay. Congrats, Larry. Okay, now, guys. Sunset cruise for 2 on Lambada from Aquamania Adventures. Ooh, mix it up.
Ava DeVries.
Woo! Sunset cruise for 2 in St. Martin.
All right, our next prize, hot glass classes. What? Oh, sorry. 3 free nights. Bring your friends and stay longer at Flamboyant Cottage, Larry and Sherry's place. 2 bedrooms, 2 bathrooms, and a pool, a beautiful pool with a 4-island view. All right, shake it, drum roll.
Marcia Stocky.
It's Dutch.
Yeah.
Okay, our next prize, hot glass classes from the Master Joe Bean. Francis Hassel from the Life Center. There's 2 drawings. Okay. Sue? Just says Sue? Oh, what? No, I thought there was 2. Yeah, there's 2. Okay. Sorry. So 2 free Wells Bay pens from Saba Finds. So we have 2 pens, and Sue got the first one. Awesome. Wanna do it? Oh. Yeah. Okay. How do you read that? Francis what?
Oh, Francois.
Francois. Yes,
Gia
yeah.
Cologne, yeah.
Gia Cologne. Okay. All right. Okay, our next prize is organic gardening class for up to 10 at Saba's farm, thanks to the Saba Reach Foundation. Carrie Smith. Okay, our next prize is indigo dyed keepsake from Anna Keene.
Tim Hammer. Tim Hammer.
Okay. Sorry. Okay, our next prize is a handcrafted quality kitchen knife with a beautiful American walnut handle from Triton Saba. All right. Thank you, John, for making that. Okay. Lynn. Oh, Lynn Lewis. Lynn Lewis. Some people's handwriting. Oh, sorry. Okay. So our next prize is a guided hike for up to 6 people from the Saba Conservation Foundation. Thank you.
Carrie Smith.
Carrie Smith. Yeah, it's on the back. All right, our next one, we're right here. Dinner for 2 at the Hideaway with your hosts Tanner and Danielle. Yeah, sorry. All right. Christina. Angelina's. All right, our 2 last prizes of the night. 3 nights, but you're going to want to add more. Not only the most amazing views, a beautiful 2-bedroom, 2-bathroom, 3 nights at our wonderful Spyglass Villa. Drum roll, please. Vito C? All right, and our last prize of the night, our grand prize. Drum roll, please. 1 week of unlimited diving, food, and fun aboard the Caribbean Explorer, part of the fleet of Explorer Ventures with liveaboard dive boats around the world. All right. Woo, who's it gonna be? Rachel Robson. Congrats. Oh, Oh, and she's a diver. Wonderful. Oh, yeah. Okay. So we just wanted to say thank you to everyone who participated or followed our events throughout the month of October, and thank you to our generous sponsors who continue to be excellent and support our program every year. So thank you so much. Thank you. Good night, everyone.
Miami is known as an international community and the hub of trade and logistics for the Caribbean and Americas. A leader in the freight logistics industry, Caribtrans has served the Caribbean region for more than 30 years, with a footprint that has grown from one island to now serving the majority, as well as Central and South America. Caribtrans is an NVOCC, a non-vessel operating common carrier, meaning it has the same responsibilities as a shipper without owning the vessels or planes. Its primary customers, individuals who ship clothing, electronics, and other personal items, and occasionally cars.
Most of the stuff that they're sending is because they don't get it there. So for us to provide it on two services, air and ocean, we can give it a choice of how fast you want it. So if you want it really fast, we're going to send it by air. You're going to have it next day, sometimes even the same day.
That on-time service has been the key to Caribtrans' success, whether shipping by air or by sea. The company moves about 5,000 TEUs of freight each year, more than any NVOCC in the region. And since it joined the Saltchuk family of transportation companies a couple of years ago, Caribtrans can offer customers even more shipping options.
Other MVOCCs, they depend on other shipping lines that are not part of their family. So they need to rely on whether they're going to sell or not, whether they are late or not. It doesn't happen to us because it's part of our family of companies. We control the service that we give.
Sí, buenos días, por favor, con Diana.
Quality service is what keeps customers coming back. Customers are treated like family, and when they leave to research competitors, it doesn't usually last long.
They have left and they have come back because they've said that, nobody does it better than Caribtrans. So because of that relationship.
That exceptional service includes consolidation. Customers can have freight sent here and stored for free for up to a month, then consolidated and shipped on to the islands and Americas, a perk for the region's growing small business market. Puerto Rico is an expanding market for Caribtrans, about the size of Connecticut and rich in history and culture. Puerto Rico is home to many pharmaceutical manufacturers, as well as a thriving tourism industry for which Caribtrans moves raw materials and supplies. But serving small businesses is still the company's bread and butter here in San Juan. Businesses like Caldera Café, a local coffee company. Owner Alfredo Rodriguez started growing coffee beans at his mountain farm about 20 years ago. He recently ordered a variety of new equipment for his farm and shop. He uses Caribtrans because it consolidates shipments, which cuts down on the number of times he has to pay the Puerto Rico import tax.
Not many companies want to do that. They want only big cargoes and maybe half container or full containers, but they don't want to consolidate because it's more difficult.
Turning challenges into opportunities is what Caribtrans does, and that includes helping to grow small businesses like Caldera Café by taking the hassle out of shipping.
We can be a gateway to expand their business in the Caribbean and back to the U.S. And wherever they can sell their products, we are also going to be a partner for them.
That customer dedication has made Caribtrans what it is today. Now as part of a larger family with more transportation resources, Caribtrans hopes to grow beyond its current market, offering services to additional Latin American ports and eventually Europe and Asia. But even when it becomes a global logistics company, Caribtrans wants to stay true to its roots, always providing personalized, reliable customer service and a commitment to the communities it serves.